Spliceosome-mediated RNA transplicing, also known as SMaRT, is an RNA-based technology that was developed in the early 2000s. To understand how SMaRT technology works, let's first recall a few concepts. DNA contains both exons, which are the coding regions, and introns, and DNA is first converted to pre-mRNA through transcription. During this process, or co-transcriptionally, RNA processing takes place. RNA processing is a series of events that convert pre-mRNA into its mature form, RNA. This is done with the addition of the 5' cap as well as with the cleavage and extension of the 3' end by poly-A polymerase, or PAP, to add the poly-A tail for stability and protection. Another step in RNA processing is RNA splicing. RNA splicing involves the removal of the introns in the pre-mRNA by spliceosomes, denoted by yellow stars in the figure. After splicing and exporting the mature mRNA transcript from the nucleus into the cytoplasm, the mRNA can be translated into an amino acid chain and further folded into a protein. There are two types of RNA splicing. Cis-splicing is when the intronic removal process occurs within the same pre-mRNA molecule, while trans-splicing is defined as splicing between two separate pre-mRNA molecules. SMaRT technology uses the natural trans-splicing process to reprogram genes for diagnostic and therapeutic purposes. This technology can repair mutations, alter specific parts of a transcript, as well as modify protein function. This is done with the use of RNA trans-splicing molecules, or RTMs. The first step of SMaRT is the initial delivery of the RTM. A RTM is a designed RNA sequence engineered to specifically bind to a target pre-mRNA and splice itself into that pre-mRNA. The RTM does this by pairing with the intron next to the mutated segment of pre-mRNA. This RTM contains 3 domains: a binding domain, a trans-splicing domain, and a coding domain. Shifting to the pre-mRNA view, we can see that once bound, the RTM prevents spliceosomes from including the mutated segment in the final spliced RNA product while replacing it with a corrected sequence. Once the RTM is bound to the target RNA, the next step of the SMaRT technology process is the continued delivery of this corrected RTM to replace the mutated piece in the final mRNA product. This technology can allow a researcher to replace and repair a 5', 3', or internal gene region depending on the target region of the engineered RTM. Step 3 is the translation of repaired mRNA to produce the unmutated functional proteins. Some strengths of SMaRT technology as a gene therapy includes that it only requires the delivery of one component not naturally present in the target cell, which is the engineered RTM. In addition, numerous mutations near one another in the same gene can be repaired using only one RTM, which provides a large benefit over CRISPR and other approaches. We also see preservation of the endogenous regulation of the target mRNA expression, as well as the dual capacity of reducing mutated protein synthesis and promoting normal protein synthesis simultaneously. Lastly, this approach relies on a small restorative molecule instead of a full-length cDNA. Here we see 3 previous studies that have used SMaRT technology to correct mutated genes correlated with genetic diseases, although many other studies have taken advantage of SMaRT. Future applications include genetic diseases that result from mutations in truncated proteins in the genes associated with the disease, with some examples listed here. In summary, exon replacement by SMaRT can repair abnormal pre-mRNA associated with a genetic disease. Here are my sources. Thank you for listening.